Bonjour à tous. Hello everybody. I am Marie Devine from the Laureat in Nancy. And I would like first to thank the local JOBIM organizing team for making it possible to have this local event here in Nancy because it is so nice. I also thank our keynote speaker, Herrmann, for making his way from Heidelberg to Nancy and to give his talk in a presential mode. So, Carl, you are a full professor in bioinformatics at the University of Heidelberg since a few months. Congratulations. And you were trained in France in applied mathematics at the École nationale des ponts et chaussées and in theoretical physics at Centre de physique théorique de Marseille. You obtained your PhD thesis in theoretical physics from University Aix-Marseille in 1999. And apparently, from what I read, it is during your second postdoc at the University of Turin that you switched it to bioinformatics, and in particular bioinformatics of co-expression networks. And then you went back to France as assistant professor in bioinformatics at the University Aix-Marseille for several years, before moving to the German Center for Cancer Research in Heidelberg in 2013. And in 2018, you created and led the biomedical genomic groups in the Health Data Science Unit at the Medical Faculty Heidelberg and BioQuant Interdisciplinary Research Center. Okay, so you have been involved in many prestigious studies involving multi-omic data and cancer, and your research is mainly focused on studying gene deregulation in disease. You developed novel methods to integrate the large-scale genomics data set, in particular epigenomics and also single-cell approaches, using statistical and machine learning approaches, which is very interesting, and you will talk to us today about explainable machine learning models for omics from black to gray boxes. So thank you very much, and the floor is yours for your keynote.
Thank you. Thanks very much. Merci beaucoup pour cette introduction. Merci aussi aux organisatrices, aux organisateurs pour l'invitation à venir parler ici à JOBIM et me donner l'occasion de revenir, ce que vous n'avez pas mentionné, que j'ai passé quelques années ici à Nancy, sur les bancs du lycée Poincaré. Donc c'est très émouvant de revenir à Nancy. Voilà, c'est aussi très émouvant et très intimidant d'être en direct sur plusieurs fuseaux horaires. J'ai jamais vécu ça, donc ça a un côté Eurovision, Mondovision, c'est très bien. Donc, merci beaucoup. So I'll switch to English for my presentation. I'll sit down so I can point to my screen. So, so today I'd like to share with you some thoughts and some projects we're currently conducting in the field of explainable machine learning models. And I've entitled it From Black to Gray Boxes, and I'll explain a little bit what I mean by that. So this is obviously an extremely wide field, and so I will just try to tackle some key questions, key concepts, and then go to the field of genomics, where many of these ideas that basically come from image analysis are now being transferred to omics field. So actually, this is not new. This is the first AI model of the 20th century. This is Deep Blue of the 20th century. So this was an incredible machine that was able to play chess automatically. So this is actually a puppet here, and this puppet, dressed as a Turk, was able to win against, the best chess players of the time. And so actually, it wasn't a black box; it was a transparent box, because the people who were showing this machine actually opened all the doors of this machine to show the interior, to show how it worked. It looked very complicated with some wheels and strings and et cetera, et cetera. So you could walk around, look inside and see, wow, this is incredible, right? So it's even better than Deep Blue because you could see actually what was happening inside. So a transparent box. So actually, it was a little bit complicated to understand exactly what was going on, but, basically who cares as long as the machine wins, right? So, obviously, it wasn't a fully transparent box, right? It was more like a gray box, because you might have guessed that it had some hidden doors and some hidden places where actually there was a real person looking and manipulating the Turk and playing against the machine, but it has a tremendous success. Benjamin Franklin played against, he was absolutely enthusiastic about that. I don't know whether he knew that actually it was a fake thing, but I mean, it was a big attraction, right? So this is a little bit the situation we are in now. And to explain a little bit the challenges of explainable AI, I'd like to show two examples that are a bit more up to date than this. So this is an example of a study on patient data that was using a large database of more than 2,000 patients with almost 200 clinical variables on clinical history of the patient, et cetera. And these were patients that had pneumonia. And the authors of the study tried to develop a model, a machine learning model, to predict dire outcome. So dire outcome means either predicting death within 30 days or ICU admission or severe complication, etc. And so they tried many different models to build such a classifier to predict this dire outcome. And then, training these different approaches, so a Bayes classifier, neural network, or linear models, logistic regression, etc. They, validated the performance of their model and found, maybe not surprisingly, that all these models had quite high AUC values, as shown here on this plot. But if you look here on these lines, the models that actually achieved the best performance were neural network models or different flavors of neural network models. Here, multitask learning neural network model, which had the highest performance, right? An AUC of 0.86. So this is a pretty good model to predict whether a patient will have severe complications or not. And so you could imagine now launching a clinical study using that kind of model. So before doing that, they looked a little more. So they had this best of the class neural network model, but they also had a second model that was a rule-based learning model that achieved a slightly lower AUC value, but had the advantage that you kind of understood what was going on, right? And so it was important to look at this model because the authors actually realized that one of the rules that had been learned by the model was that if a patient had an asthma history, if a pneumonia patient had an asthma history, actually he was classified as a lower risk patient, which obviously is counterintuitive and might not match what is known from medical doctors, right? So actually it seemed that these good performance were based on a fake rule. And so trying to understand what was going on, but they realized that in their cohort, all the patients with pneumonia and asthmatic preconditions were directly sent to ICU, in the absence of complication, but straight from the beginning of their hospital stay, they were sent to ICU, where they had, of course, more intense monitoring, more intense treatment, and a better life expectancy, of course, right? And so this is why in the data set, asthmatic patients were actually associated with a better outcome because they had this particular treatment to start with, right? So this was kind of a bias of the network. So of course, any doctor would tell you that without this ICU treatment, asthmatic patients with pneumonia are definitely at higher risk, right? And so this highlights one of the key idea behind having interpretable models is that you can identify spurious associations between features and output. And another interesting observation here is that this transparency, these rule-based learning models, has also a cost in terms of performance. So they usually perform slightly worse than other models. So the second example comes from the field of image analysis, where most of these methods and ideas have been implemented. And it's a classifier. You might know this model. It's a classifier to classify wolves from huskies, right? So you train your model on a large data bank of images. Some are wolves, some are huskies, and you see here out of these 10 images, 8 are correctly classified: husky as husky or wolf as wolf, and 2 are actually wrongly classified here: a wolf as a husky, and vice versa in this case here. And so of course, it's interesting to try to understand why some of these samples were wrongly classified. And this is one example, right, of a husky that was classified as a wolf. And the authors tried to understand, okay, why did we have this wrong classification? And they implemented a method, I'll come back to this a little bit later, where you can kind of highlight the pieces of the image that leads to the classification. And they realized that actually the pixel of the image leading to this classification had nothing to do with the animal, but had to do with the surrounding, right? So actually, whenever you had snow, the classifier would say, well, it's a wolf, right? So this is also a very interesting finding. Another example of gender classification where there were misclassifications, so people who were, defining themselves as female or male were wrongly classified by this machine. And the authors here use a slightly different approach to try to understand. They started from the original image and made slight modification to the image, training an adversarial model. And the result is this right image here, which is barely different from this image. But here, this right image is actually correctly classified by the model. Same here below. This is a slightly modified image with respect to that, but this one achieves a right classification. And now when you look at the difference between the image, you realize that actually the biggest differences here are around the eyes in these two examples, right? And so this is illustrating other aspects of explainable AI. So you can identify biased data sets. If all your worlds are in snowy surroundings, of course, the model might pick that. And it might allow you to design better sampling strategies, right? If you know that eyes are important, you would probably select images where the eyes are clearly illuminated and not hidden. Okay, so these are two examples, and what I'd like to discuss now is, okay, why do we need explainable AI? So I think I've illustrated already some of these aspects here in these examples. I'll come to that later. And then I'll try to answer the question, what actually is explainable AI, right? What do we understand? What do we mean when we talk about explainable AI? And then how is it implemented? And this is where I'll go more into the field of genomics, showing some examples of applications of these ideas in genomics, and also presenting some of our work in this field, which we're very excited about and currently is very active in our group. So why do we need explainable AI? I've shown some examples, but this is another very striking example for those who do image analysis. So these are all images that have been classified with a probability larger than 99.6%. And the model is absolutely certain that this is a guitar, this is a baseball, and this is a starfish, right? And these models work, I mean, absolutely accurate, and we actually would like to understand why the hell the model sees, I don't know, a freight car here in this image, but with a 99, almost 100% certainty. So we probably need to understand a little bit better how these models work. So three reasons why we would like to have explainable models, and some of them I've already highlighted here in this example. One is validating the algorithm. No? That's a little bit the thing about the ICU example. So finding spurious associations or avoid something that is called right for the wrong reason, right? So if we go back to our husky, imagine that this had been correctly classified as a wolf. So everything's fine. This is a wolf. It's been recognized as a wolf. But actually, you would realize that it's only been recognized as a wolf due to the snow around. This is probably not what you would expect, right? So this is an example of right for the wrong reason. So algorithm validation. The second reason has more to do with more maybe ethical or legal reasons. You want to audit the results and avoid automatic discrimination. So probably heard about these examples where, you know, these models have been trained on a large data set that was extremely biased towards, you know, white people, and then performed extremely poorly on colored people. So this is an example of these automatic discrimination that you would like to discover in the data set. And the second aspect is this right for explanation that's hotly debated among legal experts. whether the GDPR, the data protection regulation in Europe, actually implies this right for explanation of the models. But actually, a couple of days back, the European Parliament issued a statement saying that there's a right to receive explanations of decisions based on high-risk AI systems, right? So you cannot be subject to some black box decision. You have to be able to know why this decision was made. So these are more like, validation, control reasons, but there's a more positive reason, which is maybe we could discover a new mechanism if you would understand how the model inside is working, what kind of connections have been made, what kind of associations have been made, understanding the impact of input features. And if you look, if we're talking about deep neural networks, for example, trying to understand, the activation patterns of all these neurons inside this model, and I'll show some examples at the end of my presentation. So there's a lot of, terms that I use whenever, you find hundreds of websites on the topic of interpretable AI, and you have a lot of terms: completeness, interpretability, explainability, black box, gray box, transparent box, etc., etc. And so it's a little bit confusing because if you read these articles, you have the feeling that people mean slightly different things using these terms, and they are not well defined. So there's a very nice definition which I found in a kind of recent paper, which is the goal of interpretability is to describe the internals of a system in a way that's understandable to humans. And I think that's a very key point. It has to be understandable by humans. It has to work a little bit like we would also, make choices and classifications, etc. And there's a second statement which I think is also important to make and to distinguish from the first is completeness is something else. It's the goal of completeness would be to describe the operation of a system in an accurate way, right? So if we go back to the neural network example, completeness would mean I would give you all the parameters of the model, all the weights, all the biases, everything, all the hyperparameters, and then completeness would be achieved. You would be able to reproduce what my model is doing, but it doesn't mean it would be interpretable, right? So I think it's important to kind of keep in mind that interpretability and completeness are two different things. So how would we define? So I think there are two key points when we talk about interpretability. One is to understand the impact of individual input features to the model, right? So if you have your black box model, you have input features, you make predictions, classification, regression, whatever. You would like to know which one of these features actually has the biggest impact, right? So this is one aspect of the interpretability. And this you can achieve, and we'll discuss that in a minute, either by retro-engineering the prediction, right? So you go from the prediction and try to go back to the features. For example, by feature elimination, feature shuffling, etc., etc., you can get an idea. If I shuffle this feature, nothing happens. My model stays equally good or bad. So probably this feature is not super important, and otherwise, you see a drop in the performance. Or these feature importance could be the result of the model itself. I'll give an example on the next slide. So this is one aspect, and the other aspect is trying to understand the intrinsic rules of the model. So this is related to what I said previously. Maybe we can, through interpretability, maybe we can learn new associations, new connections between some of the features or representations of these features that will give us some insight that we didn't have previously, right? So if you think about the most simple machine learning model, which is a linear regression, so if you have a very simple model with two input features, X and Y, and you predict Z, from here you can directly read the impact of the features, right? If you normalize your coefficients, you normalize your data, you can read from alpha and beta the sign, the amplitude, and this gives you already immediately the importance of the input features. Now, if you add an interaction term, and you see that this coefficient here becomes important, becomes statistically significant, then that will tell you something about connections between the input features that already relate to the internal of the model, right? So this is a little bit how we can imagine the difference between this impact of input features and also the connections that are made inside the model between these features. Okay, so I said it's a pretty complicated field, and so different people have tried to make an ontology of interpretable, explainable AI models. So there's one nice review here, which kind of try to classify all the different approaches towards explainable AI. So starting from the problem of opening the black box problem, they say, well, I mean, there are two approaches. Either you don't have a black box, right? You immediately start with a design where everything is clear, right? For example, if you have a regression model, well, it's a transparent model, right? Or if you have a more complex model, you try to explain the black box. And here you also have different goals. For example, one would be model explanation, outcome explanation, and model inspection. So what are these three different topics? Well, model explanation would mean that you try to globally understand how the model reaches its conclusions, right? So this is this global interpretability. It could be the global importance of the feature over the whole cohort. Outcome explanation is something different. It's for one particular instance. You would like for one particular instance to understand why did it take this decision or this classification for this very particular instance. So this is more of a local interpretability compared to this global interpretability. And model inspection would be, how does the model react if now I change the input features, right? And so this is what is also called post hoc interpretability, right? So you train your model and then you kind of retro-engineer your model to try to, a posteriori, try to understand how the model worked, how important the features were, whereas here, this is what is called intrinsic interpretability, right? And we have a transparent model. So transparent models, I'll come to in a moment to some examples of those, right? So this is an important difference, this intrinsic versus post hoc interpretability. So does the model already provide all the interpretability, or do we need to determine that after the model is trained? The other important distinction is what I said previously between this local and global interpretability. So can you only understand globally the decisions, or can you say, pick an instance and say, why did it wrongly classify this particular instance? Why did it correctly classify this other instance? And the third aspect, and I will not talk too much about that, is approaches that would deliver interpretability no matter what model you have, right? So no matter what black box you have, this approach or this algorithm would give you interpretability, whereas some other approaches are really adapted to a very specific model. So I'd like to just show an example of this intrinsic versus post hoc interpretability. So intrinsically interpretable models would be, for example, decision trees, right? So if you have multiple features of different persons, weight, height, BMI, or whatever, you can train a decision tree model. And you immediately understand at each step how the decisions were made, what were the crucial parameters, what were the crucial threshold, etc. So this is a very transparent model, right? And of course, you know other types of transparent models. I've already mentioned linear regression models, logistic regression models, etc. And we might have this idea that nonlinear models are black boxes and linear models are interpretable models. So this is not totally true, and I'd like to give a little bit of an example of why linearity doesn't guarantee interpretability. So the first is also related to what I said previously about complexity versus interpretability. So if I train a logistic regression classifier to predict disease state or cancer subtype or whatever from expression data, if I have 25,000 input genes, it's formally interpretable. I can understand everything. I can see the 25,000 coefficients, but it's not biologically interpretable, right? So we have a completeness, but we don't really have interpretability, although this is a perfectly, I mean, it's based on a linear regression. Another interesting example, which we have worked a lot on, is if you look at two examples of linear methods like principal component analysis or non-negative matrix factorization. These are two linear methods for factorizing matrices. But I really like this example here, showing again from the field of image analysis the differences between principal components when you decompose the image of a face using principal components. I mean, this is principal component 1, 2, 3, 4, 5. So you kind of, recognize the face, sometimes more greenish, sometimes more reddish, but you don't really get why PC1 is different from PC2, PC3, etc. So it's not a very interpretable decomposition. Whereas if you decompose the same image using non-negative matrix factorization, you now see that the different signatures or the different components here have a very clear interpretation, right? The tip of the nose, the mouth, the eyes, the lips, etc., etc. So this is an example of an interpretable model, whereas PCA is more hardly interpretable. And so we have used a lot these property of non-negative matrix factorization to perform part-based decomposition. So you can nicely see in this type of plots where we have different here, histone marks, epigenetic features. We can define different epigenomic signatures, and they are clearly defined by positive contribution of certain features. And you really nicely see the patterns here that contribute to signature 1, 2, 3, or 4 because of this part-based decomposition property. I'd like to come back very briefly to this difference between the local and global interpretability, again using an example. So this was a study trying to train a model to predict glucose level in patients using 38 different types of, again, clinical and biological features. And so the model worked pretty nicely, and the authors actually tried 3 different models: random forest, generalized linear model, or gradient boost machine. They had similar performances, and they extracted the feature importances. Right? So this was a post hoc extraction of feature importance, where age was the biggest, had the biggest influence, gender, BMI, etc. So this is an example of global interpretability, where over the whole cohort you can see that on average, age and gender and BMI, etc., have an important impact. But actually, an interesting second step of the study was to look at the individual patients. And now to look again at some of these clinical variables, whether the patient had depression or what this smoking status was, physical activity, etc. And to look at the importance of each one of these clinical variables, but now for individual patients. And so, for example, if we look at the GBM model here, which would be the red dots, we see that this model actually highlights a group of patients for which depression seems to be a very important predictor of glucose level, right? And most of the red dots here are around 0, because for most of the patient it doesn't play any role, but for this subset of patients it plays an important role. And so this is a very interesting observation for a clinician or for a scientist, because now you would like to take these patients and try to understand why for those patients depression is a good predictor of glucose. So this is an example of local interpretability where you really measure the feature importance for each instance, right? So I'll skip briefly. So this is one of the measures that has been introduced to deliver this kind of local importance score for individual features. I don't want to go too much into the details of this, but the idea is, if you have a classification boundary that's not linear, how can you kind of understand the importance of each feature in classifying a blue sample or a pink sample? So the idea is, well, if I have an image that's here in this class, so this would be the class Labrador dog, for example, the idea is to kind of sample instances around this locally and to weight these instances depending on their distance to the original image, right? The further away, the less these sampled instances will play a role. And then to try to approximate the unknown model, right? So you have a black box model F that does this classification, and you kind of approximate these locally with a linear model. So don't want to go too much into the details, but the idea is that you try to approximate the true classifier F, which you don't know exactly how it works. You approximate it with a linear model here, and the different instances that you sample would be the original picture where you hide some of the patches, right? And the more you hide patches, the further away the sample would be from the original one, the closer, and then you kind of average here over the of these different instances, and you're trying to learn here the weights of a linear model that best approximates the classification of this unknown model F. And so this gives you the importance of each one of these patches that you have hidden in these different random instances. And so this is similar to what I've shown before with the wolf and the snow, but this would explain what contributes to classifying this image as a containing an electric guitar. These would be these pixels or these patches here. Acoustic guitar would be more the body of the instrument, because this is very typical of an acoustic guitar, and what explains the Labrador is actually the face of the dog. So this seems to make sense. And so this is an illustration of this LIME approach that was very popular and was then followed by other approaches to measure feature importance in nonlinear models. Okay, so now I'd like to come to how some of these ideas are being or have been implemented in genomics. And in genomics, well, we all know what kind of data you might feed in. So you might have some even more. I haven't put proteome data because I've never worked with proteomes. It's a biased selection here of genomic features. So you might have sequence data, you might have SNP, GWAS data, transcriptomic data, epigenomic data. So these would be some of the possible input features, and then you can train some, neural networks, autoencoders, convolutional neural networks to do whatever classification, regression, clustering, etc. So I'd like to go briefly to some examples, and the first would be illustrating how you can use sequence data in this kind of interpretable setting. So this is a very nice method that was among one of the first methods published implementing this kind of convolutional neural network idea on sequence data. And the question was, can we predict chromatin accessibility from the sequence alone, right? And so the authors use convolutional neural networks with filters to try to predict chromatin accessibility using as a training sample ENCODE data across 164 cell types. So what they did, they extracted DNA's hypersensitive sites, extracted the sequence around these DNA's hypersensitive sites, one-hot encoded the sequence here, and then applied a number of convolutional filters, several layers of convolutional filters. I don't go into the details, but in the end, they make a prediction for every site across the 164 cell types, and they predict it's accessible or it's not accessible in this cell type, right? And so you see, I mean, it's very tiny, but you can, at the bottom here, you see the predictions, and below you see the actual accessibility. So it seems to work pretty nicely here. Now, this could be a perfect black box model, but the cool idea of this paper is to say, well, we could maybe look at the first convolutional filter here because actually this first convolutional filter will kind of, as a built-in feature, will give you some weights on certain position here in this one hot encoded feature. And actually these weights look exactly like the weights in the position weight matrix. And you can kind of interpret the weights learned by the convolutional filter as a position weight matrix. And so this is a very, very interesting idea, because then for every sequence and every cell type, you can kind of look inside the black box or the gray box and then see, okay, what are the filters, the filter weights that have been learned, and transform that into position weight matrices. So once you have this kind of model, you can do all sorts of things. You can do in silico mutagenesis. I give you a sequence. The model can systematically mutate all positions. And then you can see how much by mutating a position you lose or you gain accessibility, right? And so this would be one example of a sequence where whatever you do here to this position, suddenly you have a huge loss in accessibility. And then you can really look at, okay, which kind of mutation at every position has the greatest impact, and again, interpret that as a motif corresponding to a potential binding site of a transcription factor. So this is a really very beautiful idea to predict chromatin accessibility. And then, of course, following this example, there were tons of other models, and it's a pattern recognition problem. So if you look at all these methods, you might learn some common features in the names of these methods that will tell you that this probably is based on deep learning approaches: deep bind, deep CPG, deep C, deep histone, and deep TACT. So whenever you see a method with this kind of name, you know that there is some DNN model behind, so you can predict chromatin conformation, DNA binding motif, epigenomic states, etc., etc. So there's a very interesting review here listing some of these models in briefings in bioinformatics 2021, which you might have a look at if you're interested in specifically this kind of models. Now, I'd like to focus on something I've mentioned at the beginning. We have, if we now go to these neural network model, where you have all these neurons, these complicated networks of neurons, and you have this mysterious activation patterns across all these neurons. Can we somehow understand why this one neuron here lights up very highly in one condition and the neighboring doesn't, and vice versa if you use another condition? So could we try to understand this? And so the idea is, well, here we have a very complicated network, which we don't have any meaning for, but biology is full of networks, right? The biomedical pathways are complicated networks, gene regulatory networks are highly complex networks. Ontologies are super complicated networks also. So the idea would be, okay, can we not use these complicated biological networks that we understand? We understand each one of these nodes. Couldn't we couple that to these deep learning models? And actually, this is an idea that was picked up by several studies. And I think one of the first was the group of Trey Eidecker, who in 2018 published this D-cell method, where using yeast data, they try to train a model that goes from genotype to phenotype, trying to predict cellular growth. So this is something you can do, but the cool thing about this method is that they kind of try to design this model in such a way that it's a visible neural network that would actually capture the different levels at which something can happen. So either molecular reactions or full pathways or organelles and very broad processes. And this kind of, hierarchical classification of processes into very specific and very broad processes is exactly what is done in an ontology, right? An ontology has some very broad terms, DNA repair, and has then some very specific terms that go to, way more precise and specific processes. And so they have used this template of the ontology and based and designed a network, a neural network, using exactly this template of the ontology, meaning that now not all neurons are connected to all other neurons in the next layer, but the connections follow these connections in the gene ontology. And so they've trained this on yeast data where genes were disrupted, and they have measured experimentally growth and genetic interactions to train this model here. And so this was a very nice idea. And for example, they could understand why the double mutant of PMT1 and IRE1 would lead to a defect in the cell growth. I mean, this is something that they observed, that is something that you could predict with a model, but with their model, they could really understand how this genotype to phenotype was actually wired, right? And they observed that actually it seemed that it was going through the endoplasmic reticulum unfolded protein response processes or response to ER stress, whereas other paths were not affected by this, right? So it seemed to be really due to some defects here in this pathway and not in this pathway, something they have also validated experimentally using a reported genes for ER, UPR activity, so this process here, and they showed that the more their system predicted a defect, the lower was actually experimentally indeed the activity of this pathway. So this prediction here was nicely experimentally validated. So there were further groups that have picked this idea of combining neural networks and gene ontology. One is GraphGoNet, also a very nice approach to do some classifications of disease state. But I'd like to in the last 5-10 minutes, go to some models that we have been working on. So we have focused on using autoencoders. So you might know autoencoders, they have this kind of bottleneck structure, an input layer, an output layer. You try to reconstruct, you try to press the data across this bottleneck. And what is interesting about these models is that you have this reduced latent space representation, right? So this is a very classical autoencoder. Now increasingly variational autoencoders are used where the latent space are not single valued nodes, but are distributions. So you learn the parameters of a normal distribution. And so at every node, you have a distinct distribution. And this is a generative model where you can sample new data point as soon as you have learned here the parameters. So this is not very interpretable, but it works nicely. So it's been used a lot in single cell, for example. So you can train such a model using single cell data. It's an unsupervised approach. And when you take your latent space, this reduced representation, and you do a UMAP or a t-SNE or whatever you like more, you see a very nice separation between the cell types, right? So this is an example of a model that works very nicely to separate some cellular phenotype, but you don't exactly know what this latent space represents. So it's unclear how this very nice separation was achieved and which one of the inputs features actually explains the difference between, for example, these two cell types. And so the idea is, again, the same to say, well, we have networks here in this autoencoder. We have an encoder and a decoder. Maybe we could, replace the decoder by something that's a biological network, right? So that's interpretable, where we would know, oh, this is the node, blah, blah, blah. This is this process. This is that process. And see if we can train this kind of hybrid models here. So you can use several things. You can say, well, my latent nodes would be pathways and they're connected to output genes. It could be transcription factors to target genes. It could be ontology terms where you have this multi-layered structure, right? And the advantage or the idea behind that is that you could then interpret the activities as biological in terms of biological processes. So one of the models implements this idea is the Vega model, where you have a latent space representing transcription factors, and you have a linear decoder with just one layer connecting the transcription factor to their known target genes, right? So you have this prior structure here in the model, and you can train this model to learn here the activities of the transcription factor. So we've used this in single cell data. So this was a data set that we have published a couple of years back of adrenal medulla, where we have identified different cell types here, precursor cells, differentiated cells, chromaffin, and neuroblasts. And we know this data set pretty well, so we're asking whether this kind of neural network would be able to capture the activity or the importance of transcription factors. So we focus on chromaffin cells and compare them to precursor cells. And now we've trained this kind of model using these two cellular populations. And then you would hopefully see a different activation pattern in this cell type or in this other cell type. And indeed, when we represent this as a violent plot here, every dot would be a transcription factor. You would see here the ones that have a higher activity in one cell type or the other cell type as actually issued by this neural network model. You actually see that the ones with the highest activity on one side or the other indeed correspond to the ones which we have experimentally determined to be important in these precursor cells here or in these differentiated chromaffin cells. So this model works very nicely. And now what we're working on is trying to see if this model can learn new connections, right? Because so far you need to provide the connections of the transcription factor, and this you take from databases or some other sort of evidence, but maybe the model could learn itself the connections, right? So we've implemented a regularization strategy to allow the model to build in some new connections, but it has to pay a price, because if it's for free, it will connect everyone to everyone. So you would have a fully connected model, but here we kind of limit the number of the connections using some regularization. And so to test this, we took one transcription factor, remove all its known targets, and looked, okay, is the model capable of recovering these known interaction partners from KLF6? And if we rank all possible connections in the model, we see using these recovery curves that actually it can recover the known KLF6 targets. This is for one particular regularization. So this doesn't work very well here. But if we go through different regimes of this regularization, a very small, very cheap regularization, a very expensive regularization, we see that there is a regime where the model kind of indeed recovers very clearly the known targets of KLF6. And this works nicely for other transcription factors. Do I have 5 minutes, or I don't know exactly when we started?
Yes, 5 minutes.
5 minutes? Okay,
yes.
Good. So I'd like to finish very briefly with another idea, where now we have used the idea of building a decoder based on a biomedical ontology, so gene ontology, phenotype ontology, etc. So this is something we've just published in bioinformatics. It's called OntoVAE. Well, again, the idea is that if we have an ontology, we in principle know the meaning of each one of these nodes here. And using GTEx gene expression data for different tissues and looking at the activation of two of these nodes corresponding to two processes, for example, digestive process, glutamate receptor process, we looked at the activation of these two nodes across all the samples on the different tissues. And what we see, for example, is that the highest activation for this node, glutamate receptor, is actually for the brain samples, right? And this works nicely with some other examples showing that we can indeed have an interpretation of these nodes in terms of biological process. So I'll skip this because of time, but I'd like to show what we can do with this kind of model. What we have done is a gene-centric perturbation where we say, well, if we have a trained model, now we can knock out a particular gene and see, okay, which processes seems to be impacted by this knockdown, or we can do the reverse. We can look at a particular process and systematically knock down all the genes here, bioinformatically knock down all the genes to see which one of these genes has the highest impact on this process. So I'd like to very briefly show one example where we knock down a gene, DMD, that's involved in Duchenne muscular dystrophy, and then looked, okay, which one of the processes seems to be mostly affected, comparing knockdown with wild type. And actually here we find all the terms we would expect related to muscular dystrophy, muscle system, but also actin filament bundle assembly. So some very specific muscle processes that are known to be impaired in dystrophy. Last slide, or almost last slide. Now, just to show you what we did also with single cell data using this kind of model, we used a data set of PBMC cells with control cells here in purple and interferon stimulated cells here in red. And so we wanted to know if our model can predict the effect of interferon on the PBMC cells. So we used CD4 T cells here in this data set to train our model to recognize the effect of interferon. What we did is simply to overexpress all the genes that we had in our input layer and trying to check which one of these genes has the highest impact here on this interferon node in our ontology. And so when we rank all the genes according to the effect on this interferon node, we see that we recover some well-known genes related to interferon. And what is important and very striking is that a lot of these genes actually are not directly annotated to the interferon process, right? Because you might think, okay, if a gene is annotated to interferon, I knock it down, of course it will have an impact on the interferon node, but actually even genes that are not directly connected to the interferon node have an impact. These would be the genes without this little square here. So this seemed to very nicely be able to predict the importance of genes in driving the interferon response. And a last plot to show why this multi-layer decoder is interesting. You have all these different terms that are activated in our decoder, each one representing a process. And if you compare, for example, blood samples to spleen samples, looking at the same part of the decoder, you see some very different activation patterns. And you see, for example, that in both B cell, here this B cell related term is highly active, but you see if you go up that it's being activated through very different pathways, right? And so this is where you see the advantage of this multi-layer representation in our model, where you have all these hierarchies of different processes. Okay, so what's next? I'll skip. I'll explain that next time, if there's a next time. And I'll skip to the conclusion slide. So interpretability is again trying to describe the internals of a system that's understandable to us, that we can really understand what is happening. We have either this gray box approach where we kind of retrospectively try to understand the input features, or we can try to build transparent box models where everything is clear from the very beginning. We have this post hoc versus intrinsic interpretability difference, which I've highlighted. And now coming to this omics applications, I think that these neural networks model can be very nicely combined to biological networks. And it's a really attractive approach to build in the interpretability and to have some transparent box models, right? And I've briefly highlighted that you can also try to make these models learn new connections or context-dependent connections, for example, connecting genes to functions or transcription factor to targets. So these are the people in the group. I've highlighted the ones who have contributed most to these topics. Daria has built this very nice onto VAE. You can check the manuscript, the GitHub. Niels, Hannah, and Anna are also working on related projects. Thanks very much for your attention.
Thank you so much. It was fascinating. I lost the eye on the chronometer because I was so fascinated. Yes, if this microphone is maybe not so strong. So thank you very much again. So are there questions in the chat? My assistant?
Nothing. No.
Sorry? But they should use the Q&R, I think. Okay. Okay. Does someone want to ask a question directly, or? What? In the room?
Well, if not, it's my role to ask some questions. So, in fact, I need maybe some clarification about the last variational autoencoder systems with this ontology, because you said that variational autoencoders are unsupervised learning because you learn the latent space representation. Yeah. And do I understand well that you first learn the latent space representation and then you replace the decoder, or is it
it's all learned as
you learn the latent space representation with this hard-coded ontology.
Absolutely.
So then it becomes rather supervised, or?
Well, it's not supervised in the sense that we're not trying to do a classification, right, in terms of cell types, etc. So the only constraint is we're trying to reconstruct the gene expression profile, and then we're seeing what's happening inside, right? Which one of these terms seems to be highly activated or lowly activated. And if I just go back to this one plot here, sorry. We see that here very clearly, in a very unsupervised way. So we haven't put any information about the tissue, but in a very unsupervised way, the model has learned that actually all the samples that come from brain seem to particularly highly activate this node, which we map to the glutamate receptor signaling pathway and digestive system process for all the samples coming from stomach, etc. So it's unsupervised, that we never put any information about this is brain, this is stomach, this is it's been learned from the model, right?
Okay, thank you. So I give the word to Nice. Was a question?
Yeah, can you hear me?
Yes.
Yes.
Okay. Hello, Carl. This is Anaïs. I think I already asked you, but I cannot remember. You systematically put your transcription factor target, your constraint, your gene ontology on the decoder layers and not on the encoder layers. So I was wondering if it would lead to the same results, and if you could also do both, like put some transcription factor target in the encoder and the ontology on the decoder or something like that.
Yeah, I mean, that's a question that was asked by the reviewer, might be you actually. So it's true. I mean, in principle, we could have built the ontology in the encoder. We actually did, and we tried. We realized that it led to slightly less reproducible results. So I mean, all of these models, because they are generative models, they have a certain level of stochasticity, right? When you run them 2 times or 3 times, you never get exactly the same result. And so we looked at the variability of these models if we would build the ontology in the encoder and the decoder, and we realized that actually having the ontology in the decoder, because it's a linear model, actually leads to more reproducible results, more stable results, probably because it's good to have first this nonlinear encoder that kind of captures the complicated nonlinear relationships, and then going through this linear decoder, right? So we're not fully sure to understand why there's this difference, but we realize that having it in the decoder. So actually, you're right, Anaïs, in the sense that sometimes you would have the feeling that the natural flow of information would rather be in the encoder, that you go from a high dimension to a low dimension. Yeah, I mean, we're working on that and we're trying different architectures. Yeah, but that's definitely a possibility, absolutely.
Another question from Nice here. So the ontology for the second model is on input, but it's not part of the model. So you cannot correct gene ontology with it, because gene ontology is, of course, prone of error, many errors. So you cannot correct the ontology with this kind of model. And second question, how can you, in the future, with new GPT model and stuff like that, with billions of parameters, how can you interpret billions of parameters?
Okay, so I'll ask the first question and hope that I won't have time to answer the second question. I mean, it's a very valid question, whether our model can correct gene ontology. So we're actually working on that. I haven't shown this, but we're working on that in the same way that we are also working on having models that kind of learn new connections between transcription factors and genes. In the same way, we're training models which might learn new associations between functions and genes. So of course, the search parameter, search space is huge, because if you allow the model to connect every gene to every term, it's going to be a very tough problem. But we are trying to focus on known multifunctional proteins. And what we're trying to see is if in different conditions, using different cell types from single cell data sets, if we see that some of these connections between a protein, a gene, and some of the functions become stronger in one context versus another context. So if we can see this effect of the multifunctionality of a protein, which is context dependent. And so in principle, you could imagine allowing the model to drop certain connections, right? And it would simply mean that this gene in this context doesn't have this function. So that's the hope we have in working on these models, but it's an absolutely valid point, yes. And the second question is, I don't know how we're going to do that, sorry. Yeah, I mean, that's why we were trying to simplify the problem. I mean, in principle, in our model, you could have a super complicated encoder, but then you go to a very simple, understandable, interpretable decoder, right? So, we could imagine a highly complex encoder. As long as you have a part that you can interpret, we hope that it will make the model still interpretable.
Thank you. And there is a question in Tours. Do you hear us, Tours? Can you speak? We don't hear you.
Je disais qu'il n'y a pas de question à Tours.
Il n'y a pas de question. No question. Okay, okay. And just you elegantly skipped all the technical programming aspects, of course, but I wanted to know if the fact to access to specific nodes, is it easy in a standard deep learning package in Python, or is it something you had to engineer yourself?
Yes. So this is something we have to engineer to kind of record the activation patterns of all these nodes. Another challenging implementation is we don't have any longer these layer structure in the decoder, because you can kind of a connection from one layer to the second next layer. So this is Daria, a PhD student, has done a tremendous job to implement this in a very flexible way. So actually our package allows to use different types of ontology, gene ontology, phenotype ontology, pathway ontology, etc. In principle, you can adapt our model to any type of ontology, and all the tools are provided in the GitHub repo. So it's a bit of development indeed, yeah. But it's, yeah.
So let's join me to thank you again.
Thanks very much.